Hello and welcome everyone to this podcast on G protein coupled receptors. And we're going to call them GPCRs for short. This is an incredibly important family of proteins. So I want to begin by talking about the GPCR family. All the proteins that function as G protein coupled receptors. And it's a big one. It's the largest family of receptors. Humans have about 700 GPCRs. Mice have more than 700. In fact, of all the ones mice have, 1,000 of them are involved in the sense of smell, which makes sense. Being able to smell various odorants is essential for the mouse to survive. We don't need quite that many. Now, I wish my kids had about that many. That way they would be able to tell me why their rooms smell the way they smell. But we'll just stick with what they were born with. They can accept a variety of signaling molecules. Almost any kind that we've talked about before, but certainly proteins, peptides, fatty acid, and amino acid derivatives, and hormones. Because they're involved in so many different pathways in a cell, they are targets for drug development. This is highlighted by the observation that 1/3 of all drugs used today, legal drugs used today, act through a GPCR. Finally, I'm going to say all GPCRs have a similar structure, which I'll draw on the next slide. But the key part is they are one polypeptide. So one gene makes one protein that makes the GPCR. The other point I want to make is that this polypeptide inserts into a membrane. I'll draw seven here. 7 pass. This polypeptide passes the membrane 7 times, and I'll draw that on the next whiteboard. Okay, so let me draw our ever so famous membrane here. And here's where I'm going to draw the G protein coupled receptor. We have one end of the polypeptide here in the extracellular space, and the other is on the cytosol side. Now it's a G protein coupled receptor, so we should probably introduce the G protein. And first, let's define what a G protein is. It's a membrane bound Gtp binding protein. It's involved in intracellular signaling. And lastly, a G protein contains 3 subunits. So let's draw it right here. The first subunit here is the alpha subunit. And you can see that it's attached to the membrane by a lipid tail. So here's the lipid tail right here. It's bound to the other 2 subunits. And so the other 2 subunits are called beta and gamma. The gamma subunit is also attached to the membrane using a lipid tail, shown here. And the beta subunit is not attached to the membrane, but it is covalently bound to the gamma subunit. As long as these 3 subunits are bound to each other, as shown here, though it's not abundantly clear, I'm trying to imply that there is this covalent interaction between all 3 subunits. So this G protein as drawn is inactive, just as the GPCR. So how are we going to activate it? Well, let's draw that down here below, so I need to erase our definition here. So let's redraw our membrane. And what activates the GPCR will be this signal here. And so let's draw what the GPCR will look like once it's activated. So we'll have the signal bind here, and I draw the GPCR slightly changed, because when it binds, it does cause one of our favorite things, a conformational change. This conformational change will allow the alpha subunit of the G protein to interact with the GPCR. And I should have mentioned, pretty big fail on my part here, that the alpha subunit in the inactive form is bound to Gdp. And remember, when G proteins are bound to GDP, that's a signal that it's off. So that's why it's inactive. And so it will, of course, bring along our gamma subunit here and the beta subunit because they're attached to each other covalently. Upon binding to the GPCR, the GDP is lost. Then what will happen is Gtp will come into this active site. That does a couple very important things. It will now make the alpha subunit active, and sometimes when we talk about an active subunit, we'll call it GS. And when we talk about an inhibited or inactive subunit, we'll call it GI. This binding of GTP does something else. It releases the gamma subunit, but it's still attached to the beta subunit. Now when that happens, the beta and the gamma subunits are now active as well. Now we'll talk about this in greater detail, but what each of these will do, they will bind to separate targeting proteins in the membrane, and they will activate these membrane proteins, which will then either produce or remove second messengers. And we'll talk about second messengers in just a moment and what they are. But we've never defined what a first messenger is, and let me go ahead and do that now. A first messenger is this original signal that comes along here and starts the whole process. Often we talk a lot about second messengers, but we don't often use the term first messenger. But since I said second messenger, you might have been wondering what happened to the first. So that's where the first is. Now, how long will this be active? How long will these second messengers be produced? How long will the alpha and the beta and gamma subunits stay active? Well, it's determined by two things. I'm going to write it here as opposed to going to a new board just so we have it in reference to the pictures here. So two, we'll call them events, determine the length of signal. So how long this will be active is dependent upon these two things. How long it's bound to target protein. So how long this alpha subunit, activated subunit, is bound to this target protein, this enzyme that makes or removes second messengers. That is ultimately dependent upon the second factor, which is Gtp hydrolysis. Gtp hydrolysis is the key switch that will turn on or off this system. So we often think of this alpha subunit as being the subunit that plays a critical role in determining the length of this signal. Once Gtp is hydrolyzed, it will then bind back to GDP, and that beta and gamma subunits will bind back to the alpha subunit, making it inactive again. So remember when we talked about those switches before, this is still the switch, it's just drawn a little bit differently. So inactive off, binds GTP, it's now on, it targets these specific enzymes. To turn it off, we hydrolyze GTP, it binds GDP, allowing the alpha, beta, and gamma subunits to once again interact with each other and keeping it off. As I look at this picture, I want to make one point before I erase it, and it's too late. The way I've drawn it, it almost looks like alpha subunits and beta gamma subunits are targeting the same target protein. That's not what usually happens. Usually they have different targets. And so I should have drawn it exactly like that, but I didn't have the room. But in reality, there would be another target protein here that the beta and gamma would activate. So interestingly, you end up having different targets for both groups here. Okay, so now let's spend some time talking about second messengers. Okay, so let's begin with a brief definition here. These are small intracellular signaling molecules that are generated or released in response to an extracellular signal. Just like we had on the last slide, we showed how the G protein, once activated, would then activate a target protein, which would then help generate or remove these small intracellular signaling molecules. Some examples include cyclic AMP, cyclic GMP, inositol, triphosphate, calcium, and diacylglycerol, which we often just refer to as DAG. So in a very general sense, this is what happens. We'll have our G protein here, which is bound with GTP, so it's active. It will interact with this target protein. And so when it interacts with it, this target protein, this enzyme, it's an enzyme, is now active. And being active, it can convert a substrate into a product. And in this case, the product will be a second messenger. This second messenger now acts as an intracellular signaling protein or molecule. So I think this will be a convenient place to stop the first podcast. And so in the next podcast, what we're going to focus on will be describing 3 of these pathways that generate second messengers. And the 3 second messengers we're going to focus on are cyclic AMP, inositol triphosphate, which will also produce DAG, and then calcium. All right, if you have any questions over the material in this podcast, please let me know. If not, I'll see you in class, and I'll see you on the next podcast. Bye.